Begin. Are there any questions? Can somebody confirm that you can hear me?
We can hear you.
Thank you.
Um, the final that's opening up next Wednesday, is there a certain time frame that has to be completed in, or is that just at any given point in time on Wednesday you need to complete that final?
You need to finish it by 11:59 p.m. It'll close at 11:59 p.m. even if you're in the exam. But you can take it anytime, starting I think 11:59 p.m. Tuesday night to 11:59 p.m. Wednesday.
Thank you.
Okay. Any other questions about the final? All right. Can you see my screen?
Yes.
Okay,
Yes.
great. So today is March 11th. Let me scroll down a little. We're supposed to be starting chapter 7. We're not there yet. We still have to finish chapter 13. I hope to finish chapter 7 today, but I don't think we'll make it. We'll see. Um, the final will be Wednesday, March 19th. You must submit it by 11:59 p.m. It'll have heavy coverage of the chapters we have not been quizzed on. So chapter 7, chapter 13, chapter 14, and lab module 9. It'll also have a few questions from the unknown project on it. It is a comprehensive final, so you can find questions from the start of the term, meaning chapter 1 through chapter, I don't know, 7. It does have questions from the lab and the lecture, but about 50% of the questions will be on chapter 7, chapter 13, chapter 14, and lab module 9. These are the material you've not been quizzed on. And then the other percentage will be on the unknown project and the previous uh, labs and lectures. Any question about any of that? This exam will be in 2 parts. Let me get here. I guess I can close it. Oop. There will be a final A, which it is timed, but you have a whole hour to work on it. And I think there's only like, it's something like 3 to 5 questions. They are fill in the blank and short, what do you call it, short essay. Generally only a sentence or two for the essay ones, but you can make a whole paragraph. It does require Respondus Lockdown Browser and the webcam. And like I said, it shouldn't take you an hour. It should really only take you maybe 10 minutes or less, but I've given you an hour to work on it. There will be a question, I think, that is like a case study, and you'll be given the information and you'll be giving tables, and you should answer the case study. But there are a few short answer and essay questions. That one is only worth 9 points, so I think there's actually only 3 questions on it. The final B is the multiple choice, multiple guess question or exam, and that one, 141 points on it. I don't remember how many questions, but it's a large number of questions. You have it's not showing me the how much time you have there, but
On that one, it says 50 minutes.
all right, thank you. So that means there's close to 50 questions. Oh, there it is there. I didn't see it. Thank you. Um, all right, any question about that? So there are two finals, final A, which are only a few questions, and final B, which are most of the questions. Final B is the timed exam where it's a multiple choice. Any question about any of that? All right, let's move on. I got some grading done. You'll have to take a look at the grade book to get that. Let's finish chapter 13 and hopefully start and finish chapter 7. We probably won't finish chapter 7 today. I've got it up here somewhere. I just need to find it. There we go. So we had talked about family retroviridae, the retroviruses, talked about how they attach, and they are unusual. Let me blow this up so you can see it better. In that when they infect a cell, they are a positive single-strand RNA virus, and they use reverse transcriptase to make first single-strand DNA, and then the reverse transcriptase works on the single-strand DNA to make double-strand DNA. Your book doesn't talk about that step. And then the double-strand DNA can be transcribed to make positive messenger viral RNA, which can be translated into the capsid proteins, which then get together with the newly replicated single-strand RNA to make the new progeny virions. And in the family Retroviridae, it is a budding virus, and so it will leave the cell by budding. And looking at that in another detail, the virus attaches to the cell, gets inside the cell. The viral RNA is made by reverse transcriptase into viral DNA. The viral DNA then integrates into the host cell's chromosome and becomes a permanent member of that cell. For as long as that cell is alive, it'll have retrovirus DNA in it. That is called the provirus. The provirus can make RNA, which can be replicated genome RNA, and that will be positive single-strand RNA, which can be read by the ribosome as viral messenger RNA to make the viral protein capsids. The virus will then self-assemble and then bud out of the, well, in the case of most retroviruses, it buds out of the cell membrane. Remember, it buds out where there'll be viral proteins and glycoproteins on the membrane, forming the new virion. Any questions about any of this? Oh, I should mention that the viral DNA integrates pretty much at random. So in different cells, it'll integrate in a different location. And that can muck up the DNA. Fortunately, humans do not have functional retroviruses in their genome. Mice do, but humans do not. We have a non-functional, actually 5 non-functional retrovirus in the human genome. And fortunately, they have mutated, and so they no longer replicate, no longer reproduce. Any question about any of that? All right, let's talk about the family coronaviruses, the coronaviridae. They are single positive strand RNA viruses. They do have an envelope. They have been known to humans a long time before COVID-19 came about in 2019. They are the second most common cause of the human cold. Most of the coronaviruses that infect humans cause human colds, but some coronaviruses do cause more serious disease like COVID-19. SARS and MERS. SARS and MERS are similar to COVID-19. SARS stands for Severe Acute Respiratory Syndrome. MERS, Middle East Respiratory Syndrome. MERS has the highest mortality rate, with a mortality rate around 36%. Fortunately, when MERS gets in a human, it does not spread from human to human. MERS tends to infect camels, and then people in close association with camels can get infected by MERS. SARS is a disease which started in China in 2003, and I think it hit the human population again a few years later. I don't remember when it was, but it's twice hit the human population. And fortunately, people got control of it and stopped it from spreading, so it's now extinct in the human population, although it does come from some animal. I think it's a bat, but they're not 100% certain where it comes from. SARS had a mortality rate of around 10%. And a good thing about SARS was it did not have asymptomatic patients. And that's why the medical profession in 2003, because it did get worldwide, it did spread to multiple continents, including North America. I don't remember if an American was infected, but at least Canadians were infected. And they were able to quarantine them and then prevent SARS from spreading. If you remember, COVID-19, President Trump, who was president at the time of the outbreak, put a quarantine on everyone coming into the United States from China. So that, and then they were taking people's temperature to make sure that somebody with COVID-19 did not come into the country. But COVID-19 came into the country, probably in an asymptomatic patient. COVID-19, when it first hit the human population, had a mortality rate of between 0.02% to 3%. That mortality rate has dropped by about 50%, so about 0.1% to about 1.5%. And I'll talk a little bit more about why that, well, it mutated and actually recombined with another coronavirus, and that's why it decreased its mortality rate. COVID-19 spreads more easily than the flu, and one person was likely to infect between 1 and 2.5 other people, and that's why COVID-19, they've never been able to get control of it. There are asymptomatic patients who then spread it around, as well as when somebody comes infected, they tend to spread at least one other person, up to 2.5 other people. Any question about any of that? COVID-19 does have a scientific name. Its scientific name is Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV 2. That name is too familiar to SARS, so most people stick with calling it COVID-19. This name is an abbreviation, CoV, or I guess CoV stands for coronavirus. It was ID, that's what the ID comes from, in 2019, and that's where the 19 comes from. So even among scientists, they tend to call it COVID-19 to distinguish it from SARS. The two are similar, meaning they are related, but they are separate viruses that cause separate diseases. COVID-19 is closely related to bat coronaviruses, and it likely originated from bat coronaviruses. In fact, they sequenced COVID-19 and they determined that it was a recombination between two bat coronaviruses and a pangolin coronavirus. The pangolin portion of COVID-19 is only about 10% of the virus, but it's important because it codes for the spikes on the coronavirus, which is why the virus is so contagious and why it's so good at spreading from people to people because of the spikes. Oops, I went forward. That's why that changed. Let me go back. There likely was an intermediate, such as pangolin, and I forget there's another animal that they think may have been an intermediate that carried the coronavirus or COVID-19 from bats to humans. Pangolin, if you're not familiar with that, is a spiny anteater. And the other animal that might have been an intermediate was something like a chow dog, which I'm not even familiar with what that animal is. COVID-19 was first seen in Wuhan, China, and they think it spread from a animal market. In China, especially back in 2019, they had these animal markets where you could buy lots of exotic animals and then take them home and I guess eat them. Was mostly what the Chinese did, uh, including pangolins they had in the animal market, other animals too. And with all the animals in close proximity and then a lot of people coming to the animal market, it's not surprising that COVID-19 moved from an animal and then infected a person. They never found the first person, but all of the first people who were COVID-19 positive had contact with this animal market in Wuhan, China, meaning it's likely that the animal carried the virus and then the animal was caught, brought to the animal market in China, and somebody bought it. Likely the animal seller or the first person who bought it likely was the first person who came down with COVID-19. The Chinese have now realized that this is a danger, having a bunch of wild animals in the animal market and having a bunch of people there, because 3 times a serious virus has spread from the animal market to the human population, meaning a virus that was not previously in humans, jumped from an animal to humans, and then began spreading among humans. This happened twice with SARS and then with COVID-19. And after COVID-19, the Chinese made a law, and so the animal markets cannot sell exotic animals like pangolins in the market. And the goal is to reduce the spread of a virus from an animal to a human, and then spreading from human to humans. Any question about any of that? All right, transmission of COVID-19 is primarily via droplets from coughs or sneezes within a range of 6 feet, but it can spread from other ways, such as especially singing can spread it, even talking. It can spread further than 6 feet, but most of the time it's within 6 feet. It can spread also from one patient touching a surface or an object, and then somebody else touches that surface and then picking up the virus. It can even spread in feces, but like I said, COVID-19 tends to spread from droplets. So it's likely breathed in, but it can be carried by hands and then to the face. It can survive up to 3 days on plastic or a metal surface, but only a day on cardboard or paper. What happens is the virus can dry out and then it can't spread. Any question about any of that? All right, COVID-19 gives mostly flu-like symptoms. runny nose and sneezing are actually now very common with COVID-19, but initially it was less common than the flu. And then shortness of breath and deaths are more frequent than with the flu. Mortality rate increases with the age of the patient, the ill health of the patient, more common in men than in women, and more common in blacks and Hispanics than in whites and Asians. The mortality rate has been seen not only in the United States, but also Canada and Mexico. So that is true. I don't know. There's no reason why it's hitting blacks and Hispanics harder than whites and Asians. There is a reason why it's hitting men harder, and that is men only have one X chromosome and they're more fragile to disease than women. So that's not surprising. And then the ill health and the age of the patient. COVID-19 is unusual in that it really hits people who are elderly and does severe damage to them. Children, even fairly young children, as long as they're, I don't know, 5 or older, COVID-19 doesn't hit them very hard. It only hits the very young, like the very newborns, where it's dangerous to them, but not as dangerous to a patient who's over 80. The actual death rate in the United States several years ago was 1.8% of patients getting the disease, getting infected, died. This number changes all the time and is going down because COVID-19 is now less lethal than when it first came about. Prevention, correct hand washing, keep your distance from people who are coughing, don't touch your face. If you're sick, you should wear a mask to prevent the spread of COVID-19. Best to stay home, keep the virus in your home. All right, any questions about COVID-19? Let me state that this picture, which is actually the picture I used for our course, came about long before COVID-19 has been around. I've been using this picture because I just thought it was a neat picture of a virus for my course long before 2019. About 10 years ago is where I discovered it and started using it for my class, maybe even longer. Any questions about COVID-19? Oh, let me state why COVID-19 is less dangerous now than it used to be, while the mortality rate has dropped by about 50%. COVID-19 recombined. The Omicron variant was a variant of COVID-19 where the COVID-19 recombined with the coronavirus that caused the human cold. And so Omicron, that variant, which was several years ago now, was less lethal. And then all of the COVID-19 variants from Omicron are descended from Omicron. So all of the variants have been less lethal. Any question about any of that? So that means COVID-19 now has recombined 4 different times. All right, let's talk a little bit about viruses and cancer. Some human cancers, about 10 to 15% of them, are caused by a virus, where the virus seems to be the first thing starting the creation of the cancer. There are human papillomaviruses, which are known to cause cervical and skin cancer, hepatitis B and hepatitis C virus, which are known to cause liver cancer. However, not all cancers are caused by a virus, like breast cancer and glioblastoma. No cases of these were found to be started by a virus. So not all cancers are associated with a virus, but some are. Cancer is uncontrolled growth of cells. The tumor cell is a cell of our body which becomes transformed, acquiring new properties. We're not going to talk about anything further than that. Viruses can cause cancer in two ways. One, you can have a formation of a provirus, and two, you can have an oncogenic virus. The formation of a provirus is where the viral DNA integrates into the host cell's chromosome, becoming a permanent change in a cell, mucking up the DNA, including mutating the DNA, turning off tumor suppressor genes, and then turning on, for lack of a better name, genes which tend to cause cancer, like an oncogene. Retroviruses, we've already mentioned, integrate into DNA, and they can muck up the DNA. They can change the expression of a gene around where the provirus integrates. And like I said, if they go right inside a gene, they're definitely going to mutate that gene. Any question about formation, how formation of proviruses cause cancer? All right. The second way a virus can cause cancer is if the virus is an oncogenic virus. An oncogenic virus contains a gene which leads to the development of a cancer. We call this gene an oncogene. It is a mutated form of a normal gene that is involved in cell growth. It's mutated so that this gene, called the oncogene, causes the cell to grow uncontrollably. And that helps transform the normal cell into a cancer cell. Any question about that? So this gene, if it gets inside your body, it will make a cell grow uncontrollably. And by itself, that's not enough to make a cancer. You actually need about 5 or 6 mutations to get a cancer. But if this cell grows uncontrollably, it'll become more than 1 or 2. It'll be a large clump. And then one of those cells can become mutated, leading to the further development of a cancer. Like I said, you need about 5 or 6 mutations to get a cancer cell, but the oncogene starts, that'll be the first mutation, starts the whole process. Any question about any of that? All right, we've talked about animal viruses having two forms, the lytic animal virus and the budding animal virus. Let me state that there are two other life cycles in animal cells that the virus may be in. There's the latent life cycle, and there's the persistent viral life cycle. The latent life cycle is a virus which becomes asymptomatic and really latent, where the virus is not replicating, not causing disease. The virus is just sitting there waiting until it leaves the latent life cycle and then enters the replication cycle, which is almost always the budding life cycle. So the virus, which is a budding virus, can go latent, where it no longer causes disease, no longer replicates. The virus just staying there, waiting. And then the virus leaves the latent life cycle and re-enters the budding life cycle, where it will become active, start replicating, start causing disease. And then once that disease goes, the virus usually re-enters the latent life cycle. The cold sores, herpes simplex virus 1, is actually a budding virus which can go into the latent life cycle. And when you actually have a cold sore, the virus is replicating, causing the disease, which is the cold sore. You are infective because you're shedding viruses. And then the budding virus can go latent, where the virus is just sitting out in the body, not replicating, not causing disease. And that's the latent life cycle. An even better example is shingles, where the virus is replicating, can spread to other people. And the first time the patient gets infected, it's called chickenpox because you have symptoms of a chickenpox. And then the chickenpox goes away, the virus becomes latent, the virus sits in a cell for the motor neuron ganglia, sits in that cell for usually decades. And then the patient with old age gets shingles, where the virus leaves the latent life cycle and enters the budding life cycle, causing disease, and the virus is replicating. And that disease is shingles. Any question about the latent life cycle? The persistent viral life cycle is similar to the budding life cycle, except that the virus is a naked virus. What happens in the persistent life cycle is the virus will replicate, usually at low numbers, and will be shed from the cell in a vesicle of that cell, meaning the viruses are put in a vesicle and then the vesicle fuses with the cell membrane, excreting the viruses outside the cell. Any question about either the latent life cycle where the virus isn't replicating or the persistent life cycle? Why this virus is similar to the budding life cycle is, well, first, it's usually shed at low numbers, similar to the budding life cycle. It is incorporated in a membrane, the vesicle membrane, not the budding life cycle is where the membrane of the cell is ripped off and then forms the envelope of the virus. This is a naked virus, the persistent life cycle, and then it's shed from the cell. The trigger for a latent virus becoming active and then entering the budding life cycle is usually a stressor. In shingles, the stressor is usually the patient's old age, ill health, which weakens the host immune system. And the virus seems to know that. And so it leaves the latent life cycle and enters the budding life cycle to replicate again. With a cold sore, the trigger is when a patient gets a cold. Why it's called the cold sore. Any question about any of that? All right. We can have another persistent viral infection, and this virus is one where the disease starts mild and the virus is replicating at low numbers, but over time the virus load increases, the signs and symptoms severity increases, the disease progresses over a long period, the virus builds up until the viral number gets very large, and it usually gets more and more severe signs and symptoms of disease. Oftentimes, the virus can never be cleared by the host immune system. It may be fatal, meaning the virus can kill the patient. AIDS, in the old days, before modern medication, the HIV virus could cause a persistent viral infection, building up over time. The signs and severity of the disease will get worse, and then the virus would eventually kill the patient. Subacute sclerosing panencephalitis, which is caused by measles virus, is another persistent virus, building up slowly over time, getting more and more load, higher and higher load, I guess I should say, and perhaps killing the patient. Excuse me here. Some persistent viral infections are tolerated, like many patients who have a cytomegalovirus infection. They can have a persistent viral infection where they have the cytomegalovirus replicating at numbers all the time, meaning generally for the lifetime of the patient. Usually with the cytomegalovirus, the numbers will build up over time, but if it's tolerated, meaning the virus gets to a certain level and then it doesn't shed more, meaning the virus builds up over time and then levels off. It's tolerated so the virus can continue replicating, perhaps for the lifetime of the patient. Others, like chickenpox, there's a few patients where the virus becomes tolerated, usually causing shingles, and then the shingles can last for the lifetime of the patient. There are some other herpes viruses which are tolerated. Any question about any of that? All right, this is looking at the different viral infections: an acute virus infection, a persistent viral infection, and then a latent infection. Remember, a latent infection will have an acute infection followed by a latent infection. So we have the number of virions on the y-axis here, and then the time on the x-axis. The acute infection is where a virus infects a patient and very quickly starts replicating. Very quickly you get a high viral load, and then very quickly the virus load decreases and then disappears, meaning the virus disappears from the patient. A good classic example of acute infection is a cold. Very few days after becoming infected, the patient gets signs or symptoms of disease, they're shedding viruses, and then very quickly the virus goes away, the patient is no longer infected. With chickenpox, it follows this pattern causing chickenpox, but then the virus goes latent. And then when the virus leaves latency and enters the budding life cycle, it starts a second hump, not as severe or as numerous as the first hump, but it can be shedding a lot of virus in the shingles. Any question about that? The persistent viral infection starts very low numbers of virus. And over time, the virus load increases. So initially, the patient might be asymptomatic, but beyond a certain threshold, they start showing signs and symptoms. And the virus increases. The signs and severity and the number of organisms increase over time. And then one of two things can happen. I guess one of three things can happen. The virus can plateau out and become a persistent viral infection tolerated by the patient. The virus load never gets higher. The second thing that can happen is the virus load can really bloom, and then the virus can kill the patient while the graph is broken there. And then I guess the third possibility is the virus can get to a certain level. It might be a high level, and then the host immune system can rid the virus from the body. So those are the three types of a persistent viral infection. Any question about any of that? Let's move on to prions. A prion is a proteinaceous infectious particle where a protein can infect a patient and then cause disease. It is known that prion disease can be inherited, it can be transmitted from patient to patient. They're usually transmitted by ingestion, transplant, like a surgery transplant, or by surgical instruments. Prions cause spongiform encephalopathy, which cause vacuoles in the brains of humans and animals. Come on. I should move this up. These three are all showing you vacuoles in the brain. That's a normal human brain. Top left. And these are human brains with prion disease where there are vacuoles forming in the brain. And that's called spongiform encephalopathy because this sort of looks like a sponge. Let's go back to where we were. In animals, prions can cause bovine spongiform encephalopathy, which is more commonly called mad cow disease. There's also the sheep disease, scrapies, and then cervid chronic wasting disease. Two prion diseases. Humans, we don't call it human spongiform encephalopathy. They're called various names after the doctors who discovered them. Creutzfeldt-Jakob disease. So most patients who get prion disease, we call it Creutzfeldt-Jakob disease. But you can also call it Gerstmann-Straussler-Schneckner syndrome after three doctors who discovered it. These signs and symptoms are different in these two diseases, and Creutzfeldt-Jakob disease tends to run in families, and then other families tend to have Gerstmann-Straussler-Schneckner syndrome. They're both prion diseases, but they cause different signs and symptoms, or at least somewhat different signs and symptoms. Fatal familial insomnia, another term for a human prion disease. And then Kuru. Kuru is the one I know the best, so I'm going to talk a little bit about Kuru. Kuru was the disease seen in the Fore tribe in Papua New Guinea. There's Papua New Guinea, and the red area is where the Fore tribe are located. When patient gets Kuru, they become unable to walk, loss of the ability to swallow or chew. They have dramatic weight loss, and eventually it leads to death. How was Kuru transmitted from patient to patient? Well, the Fore tribe had an unusual funeral ritual, which involved cooking their dead relatives and then eating their dead relatives. And when they especially ate the brain of an individual who had Kuru, the disease then spread. Any question about any of that? Now, fortunately, the wives and mothers in the Fore tribe noticed that Kuru was very frequent. And I don't know how the natives, the Fore tribe, discovered that it was from the funeral ritual, but they did. So the mothers decided, I'm not eating the flesh of my dead relative, and neither are any of my children. And so when white missionaries came to the Fore tribe, the Kuru was only seen in the males, because the men continued the funeral ritual where they cooked their dead and then ate them. The missionaries were aghast at this, and they put a stop to it, passing laws that it was against the law to eat other humans. Didn't matter that it's in the funeral ritual or the relatives, but the missionaries actually got rid of Kuru in the Fore tribe. And you can talk about missionaries as much as you want and say they're intolerant and they interfere in the cultural development of a native people. But for the Fore tribe, I have to say, in my opinion, the missionaries did a good thing for the tribe. They got rid of Kuru in the tribe. Any question about any of that? All right, I've already talked about that one, so let's move on. How does a prion cause disease? Well, there's a normal protein in a cell. It's a cell surface protein which is required on a nerve cell. And this gene is called PRP superscript C for the normal cellular gene. The trouble is PRPC can convert into PRPSC. This is stand for scabies, one of the prion diseases. And then the scabies gene can come up to the normal protein and then cause the normal protein to fold into the pattern of the scabies protein or the prion protein. So the normal protein, PRP superscript C, is the same protein, the same amino acid sequence as the PRP superscript SC or the scabies protein. The only difference is the folding pattern of the two proteins, meaning the proteins can be in a different shape. And the problem is the prion protein can replicate by coming up to the normal cellular protein and then changing that normal cellular protein into the folding pattern of the scabies protein. And then the cell says, whoops, I don't have enough of the normal protein. And so the cell makes more normal protein. But now the cell has at least 2 copies of the scabies protein, making it more likely that the scabies protein will come across the PRPC protein and then cause it to form the scabies protein. You can quickly see how this can get out of hand because the cell knows it needs more normal protein, so it makes more. And then the PRP or the scabies protein starts building up in the cell, accumulating in brain cells, and that forms the plaques that we saw earlier, as well as killing off the cells that have scabies protein in them. And that's a nerve cell, so that's obviously a bad thing. Any question about prions? Let me state that Great Britain had a prion disease. What do you call that? Blow up of prion disease because they were taking the cow brain, which nobody wanted to buy or eat, and then grinding it up and putting the cow brains on the grain of cattle, and then feeding that grain to cattle, meaning the cows were eating other cow brains, and that's how one of the cows had the scabies protein, and then that was spread to other cows that ate the grain which had been fortified by the cow brains, and now many cows had the scabies protein. They all died. The British ground up the brains, get it to grain, ground up the brains, added it to grain, fed it to more cows, and you can see how the British cow industry got an epidemic of mad cow disease. To control it, the British actually had to incinerate all of their cows and then import cattle from other countries. But that's actually what happened. During the time that the mad cow epidemic was going on, about 50 people came down with Creutzfeldt-Jakob disease, which they were almost likely to get from eating tainted beef, meaning beef that had prion protein in it. And not only 50 people, but about 50 British cats, both zoological zoo cats as well as domesticated kitty cats, came down with prion disease. Fortunately, the dogs were more resistant and they never came down with prion disease. Any question about any of that? All right, let's move on to plant viruses and viroids. Plant viruses resemble animal viruses in many ways, other than the fact that they don't infect animal cells, they infect plant cells. Plant viruses cause many diseases of economic importance, such as beans, corn, sugarcane, and potatoes. Because of the tough cell wall of plants, the virus tends to enter the cells through, or the plants, is through wounds or via parasites, like you have a nematode or an insect eating on the plant. The virus tends to infect those cells. They can also spread to the cell after the cell is infected with a fungus. And then infected plants can spread the virus in their pollen and their seeds, continuing the virus in the life cycle of the plant. Any question about plant viruses? All right, that's all you need to know about plant viruses, because plant viruses do not cause human disease. And the focus of this class is on agents which cause human disease. Plant viruses do not, so we're not going to talk about them further. Let me talk a little bit about viroids. A viroid is a naked RNA molecule which is infectious and then causes disease. Fortunately, there are no viroids known in animal cell. Viroids only infect plants. And the one I'm going to talk about, there are a couple of viroids which infect plants. When they infect a normal potato, shown here on the left, it makes the potato spindly-like, reducing the size of the potato or the weight of the potato, as well as the shape of the potato. Any question about viroids? Oh, let me get out of here. Oh, a viroid, you can think of it as a naked RNA virus minus the protein coat of the virus, meaning it's a naked RNA. It can infect a cell, replicate in the cell, take over the cell to replicate the viroid, and then be shed from the cell. So it's a naked RNA virus without a protein coat. And like I said, it's really a viroid, a messenger, a strand of RNA which causes disease and can replicate. It only causes plant pathogens, and so we're not going to talk about it, viroids, further. Any question about any of that? Any question about chapter 13? If not, let's move on to chapter 7. Chapter 7 is a chapter I always end my lesson with, because it's talking about something that students already have a pretty good understanding of, and that's the control of microbial growth. Know its sterilization, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, what's a biocide and a germicide. Know all these terms. 2, be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways in which an antimicrobial agent can kill or inhibit a cell. 4, understand the various types of disinfectants and how they work. And then 5, understand the relative resistance of the major microbial groups to antimicrobial agents. Any question about any of that? All right. Let me blow this slide up. So when we're talking about microbial death rates, bacterial populations tend to die at a constant rate if it's plotted on the semi-log graph, meaning when we have the log of the number of microbes up here, and then the time here, and then graph it on the log or semi-log scale, it follows a constant death rate where you go from one to another, and then you have a linear death rate. If you were to draw this on a arithmetic scale, where the number of survivors is on the arithmetic scale this way, and then the time is arithmetic in both cases, the curve is not logarithmic, not a linear line, and it follows an exponential curve. Any question about any of that? So microbiologists like to plot it on a semi-log plot because when we do, we have a constant death rate. And to describe this equation is very easy. It's something like y equals mx plus b. To describe this equation is much more difficult. It would be something like y equals mx plus nx squared plus b, or maybe plus b and then plus bb, something like that. What does this mean when cells die at a constant logarithmic rate? It means if we have 1,000,000 cells and then expose them to 1 unit of time, you'll have only 100,000 cells left, meaning 900,000 cells have died. So only 1/10 the cells are alive after 1 unit of time. And then you go for 2 units of time. Once again, you have a die-off of 1/10 of these cells, or 1/100th of the total population of cells. And then you have another 10-fold dilution by adding another unit of time, another 10-fold dilution, adding another unit of time, down to where you have only 1 cell surviving after about 8 units of time. Any question about any of that? So what does that mean? It means if you have a large number of cells and you want to kill all of them, you need a long time of exposure to your killing agent. Any question about any of that? All right, the microbial death rate will depend on several factors. Obviously, the number of microbes. The more microbes you start with, the longer exposure time you'll need to reduce the number of cells down to a low number. Whereas when you have a lower number, it'll take less time to reach, well, 0 or 1/1000th of the starting cells. Environmental influences can affect the microbial death rate. For example, the presence of organic matter can influence the death rate. Like bleach is easily inactivated by organic matter, and that can ruin the effectiveness of the bleach on killing the microbes. Please understand that most hospital specimens will have organic matter around the bacteria or other microbes. The time of exposure is important. Obviously, to kill all the cells, you need longer time periods. And then there are microbial death characteristics, which can affect the death rate. I'll talk about that later, the microbial characteristics affecting the death rate or the survivability of the cells. For one, there's the macolic acid of a mycobacteria, very strong outer membrane. It prevents a lot of bactericidal agents from even getting into and damaging the microbial agent. And that's just one characteristic of a cell that can infect or influence the microbial death rate. Any questions about any of that? All right. Oops, I went the wrong way. This is showing you some terminology. I'm not going to talk about it, but what's sterilization, commercial sterilization, disinfection, antiseptics, degerming, sanitization, how the terms are used. Please realize that if you're using a chemical on an object, you will then have disinfection occurring. But if you use that same chemical on living tissue, we don't call it disinfection, we call it antisepsis. Any question about any of that? For example, I could use bleach on the toilet to clean the toilet. I'd be using bleach as a disinfectant. But if my cat came home, I don't have a cat, by the way. If my cat came home and I used bleach to kill the infection of its wound, bleach would be used as an antiseptic compound. Any question about any of that? All right, some more terms. Biocide or germicide. These are substances that kill microbes. Most microbes would be killed by a biocide. It kills obviously bacteria, fungi, archaebacteria. Maybe, maybe not. It might kill eukaryotic cells too. You can, the biocide or germicide usually will not kill an endospore. We only have a few biocides or germicides which can kill an endospore. Anyone remember an example of a biocide or germicide which does not kill an endospore? Anyone have an example? Well, bleach oftentimes does not kill endospores. It depends on the species, but most endospores are resistant to treatment by bleach, but not all. There are some which can be killed by bleach. We have different names for different agents, like a bactericide is an agent that kills bacteria cells, a fungicide, an agent which kills fungi, and then viricide, an agent which inactivates viruses. Any question about any of that? All right, so biocide is the killing of cells. Bacteriostasis is inhibiting the growth of microbes. So you're only inhibiting the growth, you're not killing the cells. And once the bacteriostatic agent is no longer there, either has been removed or expires, the microbes can restart growing. Any question about any of that? A bacteriostatic agent is actually chloramphenicol, where we're relying on the host property to remove the agent from the patient because the antibiotic won't. The antibiotic only stops the microbes from growing, in this case, the bacteria from growing. And that reduces the burden on the cells, and hopefully the cell's immune system will be hot enough to sterilize the temperature, sterilize the microbes. Any question about any of that? All right, sepsis, another term, refers to microbial contamination. Asepsis is the absence of contamination by unwanted organisms. In this lab, you use aseptic technique. It is used in surgery to minimize contamination from the surgical instruments, the operating personnel, and the patient and the air. Any question about any of that? All right. Antimicrobial agents tend to kill or inhibit microbes by one of three ways. First, the antimicrobial agent can alter the membrane, killing the cell and then preventing the microbe from reproducing inside the cell or using the cell to reproduce. You can damage the plasma membrane, causing the cellular contents to escape and interfere with normal metabolism. Polymyxin B is an antibiotic which alters the membrane, putting a hole in the membrane, and then the cytoplasm leaks out, killing the cell. You can also damage the intracellular proteins with the microbial agent, where you denature proteins, such as an enzyme, by targeting disulfide bridges. Alcohol is usually damages the cell or the microbe by damaging the protein in the cell. But realize alcohol can also put a hole in the cell membrane. So I suppose alcohol could kill the cell both ways. Third way that a microbe can damage a cell is damage to the nucleic acid. Once you get the cell which is mutated, the virus cannot grow, assuming the virus needs the cell to grow, which it has to because the virus can't grow outside its own host cell. An example of that would be a DNA analog, which would muck up the DNA, preventing the virus from growing. Any question about the three ways microbial agents can stop or inhibit antimicrobial agents? All right, there are physical methods to control microbes, such as heat, filtration, low temperature, high pressure, desiccation, osmotic pressure, and radiation. All of these can prevent the growth of microbes if they do not kill the microbe. Many of them will both prevent the growth and kill the microbe. I keep on getting my mouse over here. There we go. Heat's another physical method which can be used to control microbial growth. All methods of heating kill cells by denaturing protein and enzymes, though if you're flaming the cells like they're on your loop and you put the loop into the flaming can incinerate the cells and turn them into char, much more than just denaturing proteins. You're actually changing the protein and then killing the cell. So we have two types of wet heat. I should say two types of heat. There's wet heat and then dry heat. Wet heat are things like boiling, autoclaving, steaming something. Those are all wet heats. Dry heat includes baking, like putting something in your oven, flaming, and incineration. Flaming and incineration are at very high temperatures, and they will turn the cell into char. Any question about any of that? Moist heat tends to denature proteins, like coagulating the proteins and enzymes. And here we're looking at moist heat denaturing the protein. This egg was stuck in water, and the egg whites became denatured by the heat of the water, like boiling water, forming the egg white there. Boiling liquid water or flowing steam kill vegetative bacterial pathogens, most viruses, fungi, fungal spores. And then after 10 minutes of boiling, it can kill many different microbes. Not all microbes, but many different microbes. Endospores are an exception, and some viruses take longer to destroy. The endospore take longer to destroy because the endospore has a tough outer membrane, and it's difficult for something to come into the cells and then come out of the cell. So endospores may take much longer than just 10 minutes to kill the vegetative pathogen. Most vegetative pathogens, most bacterial pathogens, most viruses, most fungi, most fungal spores will die within 10 minutes of boiling water. And oftentimes it'll be much sooner, like, COVID-19 put in boiling water or E. coli put in boiling water. The virus will decay, or the patient's food will start decaying and then taste bad. How do we do reliable sterilization with moist heat? It requires temperatures above boiling and pressure above normal standard atmospheric pressure. And that would be an autoclave, which is very similar to a pressure cooker, where you have steam under pressure, same as a pressure cooker, very effective on killing microbes. You have steam at 121 degrees for 15 minutes, and then the steam at 15 pounds per square inch. The temperature, like I said, 121 degrees. That will kill all microbes within 1 liter. Excuse me, I'm trying to think of the name, and I don't remember. So let's just say hectoliter. And then you stick something in the autoclave, and then when it's done after 15 minutes, it is guaranteed to be having no bacterial spores, no endospores, no other cells. For steam, meaning using steam, not with an autoclave, not under pressure, sterilization of the surface requires the steam directly come into contact with the microbe. So with steam, you can only sterilize the surface. You cannot sterilize deep inside with steam. All right. Louis Pasteur started pasteurization, where you reduce the number of spoilage organisms and you kill all pathogens, but you do not sterilize the agent you are pasteurizing, meaning it does have a shelf life because it does have microbes in it, even if you don't open it. And it would be a problem, and why the pasteurized milk does have a shelf life. It is close to real milk. It's not real milk because it's pasteurized, but it does have a shelf life. But its shelf life is longer than you would have with unpasteurized milk. I doubt any of you have bought unpasteurized milk because it's illegal in most states, but um, if you had, you would get the spoilage organisms, lots of microbes in the milk, and it would spoil sooner than the pasteurized milk. Pasteurized milk does have microbes in it, but none of those microbes cause human disease. Pasteurization is using mild heat to kill off organisms that cause spoilage. Please understand that thermoduric organisms, which are heat resistant, can survive pasteurization. There are two treatments for getting pasteurized milk or anything else that's pasteurized. Pasteurization is usually only done to liquids or to gas because it's difficult to get pasteurized solids, but it is possible. Like you could stick something in a pressure cooker or something, boiling pot, and keep it there. Anyways, the two treatments for pasteurization are 63 degrees Celsius for 30 minutes exposure or 72 degrees Celsius for 15 seconds of exposure. Most milk products, which are pasteurized, use 72 degrees Celsius. Milk, you can also find it sterilized where you heat it to 140 degrees Celsius. You only need that temperature for 1 second. It's called ultra high temperature milk or UHT milk. It does not require sterilization until you open it because it's sealed. It is also not pasteurized milk because it is ultra high temperature milk where you expose the milk to 140 degrees C, sterilizing the milk. This cat is showing you that of the methods for controlling microbes, that dry heat is not the most efficient. The cat climbed into the oven while it was warm because the kitty liked the warm temperature. Dry heat kills by oxidation, such as flaming and incineration, killing cells by, well, flaming really, incineration. And this would be hot air sterilization or baking, where the item is placed in a 170-degree oven for 2 hours. How you can know that dry heat is not as efficient as wet heat is we all stick our hands into a hot oven. The dry heat does not burn our hand, and that's because the transfer of heat from the oven to your hand happens slowly. Why the cat climbed into the oven. If you use wet heat, like steam, and you pass your hand through the steam, you'll instantly be burned. And that's because dry heat is not as efficient as transferring energy to our object of interest, at least not very quickly. And wet heat is more efficient and you can transfer the heat quickly. Filtration is a method for microbial control, and what you do is you pass a liquid or a gas through a filter. The filter collects the bacteria and microbes on this side, and then you have the sterile media or whatever, liquid or gas, on this side of the filter. Filters have pores in them, which block the microbe. They're useful for sterilizing liquids or gas. They do not work well for solids. Some foods which can be filter sterilized include certain juices, Coors beer, vaccines. Milk is usually not filtered because the blobs of fat, as well as maybe the ingredients they've added for pasteurization, can get stuck in the filters. All right, any questions about chapter 7? If not, I'm going to end it here and we will continue with this next time. There is no lab, but if you have questions, I'll be logging in at 6:30 to answer questions. I'll be logging out at 6:45 or until the last student question is answered. Any questions about anything? All right. I will see you in the next class. Bye.